So student, today we are going to learn about gram staining. Gram staining, as you can see, is differentiating between the types of bacteria on the type of stain that they harbor on their membrane. So the material required for this experiment is a microscope slide, clothespin, an inoculating loop to inoculate the bacteria on the slide, paper towels, and as you know, a microscope to visualize the slides. The shared supplies is crystal oil stain, safranin red stain, grams iodine, ethyl alcohol, water bottle, rinsing basin, alcohol burner, wax pencil, matches, microscope. So these are the basic utilities required. So these are the instructions to complete your experiment. So we are going to skip that as these are fairly basic and we have covered them in the previous classes. So the concept of gram staining, as you can see, is crystal violet dissociates in aqueous solutions into crystal violet plus and chloride minus ions. Now these ions, as you know, have penetration power through the cell walls of both gram positive and gram negative cells. So the crystal violet positive ion interacts with the negatively charged membrane of bacterial cells, and hence that's the reason for the purple color of the cells. Now the iodine, that is I3 negative, interacts with Cb plus and forms large complexes of crystal violet and iodine within the inner and outer layers of the cell. So as you know, iodine is referred to as a mordant and is a trapping agent that prevents the removal of Cvi complex and therefore colors the cell. So that's the basis of the experiment, and when you want to decolorize the excess stain that has been added, you add ethyl alcohol to the slide. So when the ethyl alcohol sets, it reacts with the crystal violet stain, and hence the excess stain that is not absorbed on the bacterial surface is washed off. Now, the counter staining gram negative bacteria, the stain used herein is safranin red, and the process is fairly similar. So what you do is you leave safranin red on your sample for 1 minute, you rinse with water and blot with a blotting paper or let it dry. And for viewing the bacteria, we generally use the immersion lens of microscope, which uses the 100x objective. Now, uh, what I need you to do is sketch the bacteria as you see in the microscope and label whether it is a gram positive or gram negative bacteria. In addition, I want you to identify the shape and arrangement of bacteria, like whether it is monococcus, streptococcus, etc., or is it colony, or is it a rod-shaped bacteria or a cocci-shaped bacteria, that is circular bacteria. So these are the things I want you to note down in your lab notebook. So this is a representative picture of gram-positive bacteria from firmicutes and actinobacteria. This is a gram-positive streptococcus. As you can see, it is a linear chain of circularly shaped bacterial molecules, bacterial cells. And this is a rod-shaped bacillus. As you can see, clearly the structure has been demarcated by the crystal violet stain. This is a gram-positive bacterium. This is further gram-positive staphylococcus, and this is the gram-negative bacteria. So that's the end of the slideshow. Thank you students for watching this video.